Right, ladies and gentlemen, welcome to this week's Geotalk. Thank you for joining us. I have to say it's really cool to see so many undergrad students in the audience who are doing the origins of life right now. So welcome. Thank you to everyone who's joined us online. And it's also super to welcome Sori, who is a recent, as of maybe 2 days ago, MSc graduate. And this evening you'll hear the product of one of the products of her MSc. And Sori is about to take up a PhD at the University of Arizona. So it's fantastic to have you in the audience, and thank you for agreeing to give this talk. And thank you to John Hancock's and CCIC for sponsoring these talks. So without further ado, we will open up your talk, Sori, and share the screen. Okay, it's over to you. Thank you so much.
Thank you. So first of all, I'd just like to introduce myself again. My name's Sori, and I'm a recent master's graduate and also a science communication intern for the Evolutionary Studies Institute at Wits. So, and first of all, I'd just like to thank everyone for letting me be a part of this conference and present my research, which I believe is vital to understanding how life on Earth originated and evolved, which is the role of death in the emergence of eukaryotes. So because my talk is going to be heavily based on genetics and bioinformatics, I'm going to skip the technical side of my research as much as possible and try to give an overall summary of my findings, but please feel free to ask me questions later. Okay, so I'm going to start off by telling you the conclusions first so that you know what to expect. And it's not going to make much sense now, but it will definitely at the end. So the conclusion I got from my research project was that we found genes encoding the ancestors of one of the main effectors of the programmed cell death, or the PCD molecular pathway in archaea. So as you can see on this tree of life diagram, you can see that some of the proteins that are ancestors of one of these main effectors were present in the deep branching phyla of archaean bacteria. And we basically concluded that they were present since the, prior to diversification of archaean bacteria. And this is quite an astonishing claim to make, because first of all, this suggests that certain PC genetic toolkits were present since the early microbial life, which is surprising because just a few years ago, we thought PCD was a hallmark of multicellularity. But for now, we know that PCD acts as an important mechanism for both prokaryotes and unicellular eukaryotes, which acts as an altruistic mechanism to benefit the entire population. And then second of all, we found that these ancestral PCD effectors may have been initially associated with cell survival rather than cell death, which later was co-opted to be part of the PCD molecular pathway and incorporated as a conflict-mediated mechanism for eukaryogenesis. Okay, so going back to the beginning, I'd like to set the scene as to where my research focus was. So as you can see on here, you can see the major timeline of Earth beginning from the origin around 4.5 billion years ago to the current. So we have evidence of life around 3.8 billion years ago, and we commonly refer to this as the last universal common ancestor, Thate or Lucas. And then a few period after, we have a diverse microbial life that arose starting around from 3.42 billion years ago, where diverse microbial life, bacteria and archaea started to rise. So at this period of time, we know that the prokaryotic organisms were very diverse, ranging from methanogens to cyanobacteria, as well as organisms involved in the nitrogen or the sulfur cycle, as well as living in diverse environments that varied in temperature and acidity. But then, after this period, something quite remarkable happened around this time, which is the rise of the first eukaryotic common ancestor, also called Fika. So Fika is suggested to arise during the through the endosymbiotic event, which is basically the archaeon and the alpha proteobacterium coming together to form Fika. So they were initially in cooperation with each other because of their metabolic complementation and using each other's metabolic waste. But then over time, they became codependent on each other, which eventually led to the archaeon engulfing the alpha proteobacterium and forming Vika. And then additional eukaryotic cellular features was acquired to form the last eukaryotic common ancestor called Vika. And this entire period of time is called eukaryogenesis, the rise of eukaryotes that we have currently today. So now that we have the timeline set, I'd like to explain a little bit on how programmed cell death, PCD, may have played a role in eukaryogenesis. So as shown on the figure here, it's expected that in a major transition like eukaryogenesis, this symbiotic relationship, for example, between the archaeon and the alpha proteobacterium, would have constantly gone through cycles of cooperation, conflict, and conflict mediation for them to remain in a stable union. And one of the reasons for cooperation would have been metabolic complementation by using each other's metabolic waste. But then what happened was the great oxidation event. So the rise of oxygen levels on Earth, which was a drastic environmental change for the symbiotic group. And the rise of oxygen basically meant that this aerobic alpha proteobacterium was in a more advantageous state than the anaerobic archaeon, and different degrees of metabolic waste was produced between these two different organisms, and this would have led to conflict. And it suggested that programmed cell death was one of the important conflict mediator mechanisms for the rise of Fika. So what PCD would have allowed was to promote the death of the groups that did not have their evolutionary interests aligned. So basically only allowing the symbiotic groups that could stay in cooperation with each other to survive. So to explain a little bit on PCD quickly, PCD is a genetically programmed form of cell death. So this is a generic figure showing the molecular pathways of different types of PCD present in metazoa. And the reason I'm showing you this is to show how extremely complicated PCD is and how diverse the different PCD molecular pathways are, involving multiple proteins and multiple protein networks. But the important part to take from this diagram for this research presentation is that caspases are one of the main effectors of multiple PCD molecular pathways. And what I basically wanted to do in my project was to study these caspase proteins as well as their ancestors. So coming back to this diagram for a quick recap, PCD acted as a conflict mediator during major transitions by genetically aligning the lower level units, basically only allowing the organisms that could stay in cooperation with each other to survive and killing off the others. But the exception with eukaryogenesis is that PCD is thought to originate with the endosymbiotic event. And this is because previously there was a lack of any empirical evidence of PCD in prokaryotes. And also the caspases main effectors of the PCD pathway was assumed to be absent in archaea and assumed to originate from the alpha proteobacterium during the endosymbiotic event. But there are two gaps in the statement. One being that studies looking for cas-based homologs in archaea has been neglected from PCD studies completely until now, because there was a lack of genomic information available for archaeal organisms. And secondly, PCD has been identified in bacteria, which pushes forward the origin of PCD prior to eukaryogenesis, rather than PCD rising with eukaryotes. So what I basically wanted to do in the study was to investigate presence of cas-based homologs in archaea, basically the ancestor proteins of cas-bases, which may differ in structure and function. And this is because additional sequences have been available since the previous studies with advances in sequencing and culturing methods. Secondly, I wanted to study the evolutionary history of the cas-bases and its homologs, and if there were any cas-based homologs in archaea, then where did these proteins come from? Okay, so for my methods were entirely computational, involving sequence, phylogenetic, and phyletic pattern analyses. And I'm not going to bore you with the details, but please feel free to ask me questions later. So I'd like to summarize the most important results. First of all, we did identify cas-based homologs in archaea. So as I showed you at the beginning of my presentation, this is the tree of life with archaea, eukarya, and bacteria, and all of the major phyla represented here. And then what I did was I mapped the taxonomic distribution of various cas-bases, which are shown in purple here, and cas-based homologs, metacas-bases, orthocas-bases, and paracas-bases onto the different lineages. And what I basically saw was that certain cas-based homologs were specific to the eukaryotes, whereas some cas-based homologs, as you can see in archaea and bacteria, were widely distributed among all of the major phyla of archaea and bacteria. And this included the deep branching phyla near the root here, the last universal from an ancestor state. And this wide taxonomic distribution raised new questions, which basically was, were cas-based homologs present prior to the diversification of bacteria and archaea, basically Lucas, or did they arise after the divergence of these two domains, and then it arose in one of the lineages, and it just happened to spread across different organisms via horizontal gene transfer. And how we basically wanted to study this was to look at their phylogenetic tree. So you can see on the figure 5 here that this is a phylogenetic tree of orthocas-bases, a type of a cas-based homolog. And the pink, so each label represents one protein sequence, and the protein sequences have been labeled according to the domains. So the pink is archaea, the blue is bacteria, and yellow and orange are eukaryotes. And you can see that the distribution or the topology of this phylogenetic tree didn't correspond to the tree of life as shown on the figure here. So basically, the cas-based homologs belonging to each domain didn't cluster together. And the incongruence between the gene tree and the species tree is most likely explained by horizontal gene transfer. So horizontal gene transfer is transfer of genes between organisms in mechanisms other than using the vertical transfer of genes from parent to offspring. And basically what you can see that there was a lot of horizontal gene transfer between different cas-based homologs and between different domains. But by looking at the wide taxonomic distribution of various cas-based homologs with this phylogenetic tree, we came to a conclusion that these cas-based homologs may have arisen prior to the divergence of prokaryotes, basically since Lucas. And we don't actually have proof that they arose prior to the diversification, but we claim that it was the most parsimonious explanation. Okay, so now that our study suggested that caspase homologs were present since Lucas, now we wanted to look at their ancestral functions. And this is because caspase homologs are pleiotropic in nature, which means they're involved in cell survival and cell death pathways. And this happens by the caspase homologs activating different proteins and protein networks according to different stimuli. And basically how we wanted to infer the ancestral function was through phyletic pattern analysis. Phyletic pattern analysis basically refers to looking at an entire protein sequence of a certain protein. And within a protein sequence, the sequence is divided into certain regions that is responsible for a specific function. And we call these protein domains. So if you look at the simplified protein sequence of a caspase, you can see that it has the P20 domain, which is a region of the protein sequence required for their proteolytic activity, and then the P10 domain for their regulation. But then caspases also require the death domain to be present within their protein sequence that is responsible to act as either adapters or the receptors of the PCD molecular pathway and allow PCD to occur in the organism. So basically, when we look at the entire protein sequence made up of different protein domains, we can infer the overall role of the protein, and we call this phyletic pattern analysis, or looking at their entire protein domain architectures. And so we basically wanted to do this for the archaeal caspase homologs that we found as well, to infer their function as well as their ancestral roles. And so this is the domain architecture diagram, where you have the protein sequences from beginning to the end, and then the abundance on the side here. And you can see that archaeal caspase homologs all had the P20 domain required for their proteolytic activity. But what is interesting was that they also possessed protein domains that was associated with the PCD molecular pathway. But what is more interesting was that protein domains associated with cell survival, such as the one cell differentiation, cell cycle, or cell metabolism, was more abundant in archaeal caspase homologs than the ones associated with PCD. And when we actually looked at their entire protein domain architectures to infer their overall function, we found that most of them were either associated with acting as receptor peptides to respond to the external environment or neighboring cells, as well as cytosolic proteins that is associated with a cell survival related function. And the fact that archaeal caspase homologs were mainly associated with cell survival raised an interesting question of whether these homologs were associated with cell survival or PCD at the time of eukaryogenesis. So we basically wanted to answer this question by mapping the different domains of protein domains of archaeal caspase homologs onto the tree of life. So obviously for bacteria and eukarya, it was excluded entirely because we didn't do the analysis. But then for archaea, you can see that we have the last universal common ancestor state here. And then for the caspase homologs present in the deep branching Depan phylum, they had extremely simple domain architectures, just possessing the P20 domain for their proteolytic activity. But then with the rise of other archaeal phyla, the Thermoplasmatoda and the Eukaryotes, they started to acquire additional protein domains associated with cell survival, as well as additional regulatory domains for to regulate their enzyme function. But so it's likely that at this point in time, they were mostly associated with cell survival than PCD. But what is very interesting was that for these late branching phyla of archaea, the ascot and the tac, they started to possess additional protein domains associated with the PCD molecular pathway. And it's very likely that if PCD did indeed act as a conflict mediator during eukaryogenesis, then these Cas-based homologs possessing protein domains associated with PCD may have played part of a role as a conflict mediator for eukaryogenesis. And what's really interesting about this evolutionary history is that this actually supports one of the hypotheses of the origin of PCD, called the original sin hypothesis by a Mason. So some of the questions in this field is frequently answered in a philosophical nature, and a philosopher called Jean-Claude Mason suggested PCD arose from a different combination and subsequent refinement of gene products participating in cell survival, and how all organisms on Earth have an intrinsic original sin in our genes to die. But going back to the original period of life we were focusing on, what does this mean for eukaryogenesis? So this evolutionary history has actually shown that there is an interconnected origin of eukaryotes and eukaryotic cell death. So as stated by original sin hypothesis, these symbionts were genetically, so these initial symbionts, archaea and bacteria, were genetically primed to induce death in itself and in others. And with the rise in conflict, ancestral PC genetic toolkits were incorporated into the conflict mediator mechanism for the rise of eukaryotes. But this pathway was also conserved and over time evolved into the eukaryotic PCD molecular pathway. So coming back to this conclusion slide, I hope it makes more sense when I say that and explain how important it is when I say Cas-based homologs, the ancestors of one of the main effectors of PCD, are present in archaea. And based on the data, Cas-based homologs may have emerged very early on, prior to the diversification of archaea and bacteria, and phyletic pattern analysis suggests they were survival related and later were integrated into the molecular pathway of PCD. Basically, what I'm trying to say is that some of the elements of PCD may be as old as beginning of microbial life, where organisms were genetically primed to induce death in itself and in others, which was used during conflict mediation of eukaryogenesis, and at the same time led to the rise of eukaryotic PCD molecular pathway. Lastly, I'd just like to thank my supervisor, Prof. Duran, and my co-supervisor, Dr. Glover, for their assistance, as well as the various financial institutions for funding my research. Thank you.
Okay, Sori, thank you so much. That was great and very different to what we normally listen to in this room, but that is a good thing because it gets geologists thinking.
Yes.
So that, in my view, is very, very important. So do we have any questions from the audience? Here we go. You have to speak a little bit loudly because the microphone is there.
Okay, I actually have 3 questions.
Sure.
Basically, I'm interested in what sampling method did you use? The sampling methods.
Oh, the methodology?
Yeah.
So it is bioinformatics based. So first of all, we actually had to look for sequences in archaeal genomes for these proteins. And then what we did was we first made sure that these sequences were actually Cas-based homologs by using HAMA, which is it's like a mathematical modeling tool. Yeah. So we made sure that the sequences are indeed Cas-based homologs. And then we did a lot of additional analysis, like structural analysis, and just to validate that they are Cas-based homologs. Because that was.
How can you relate the tree of life with the time scales? Because firstly, I saw that you had a time scale.
Mm-hmm. Okay.
I'm more interested in the great observation event. Since the archaea is single celled and single celled,
yes,
it cannot really live without oxygen. So what really happened when it got to a time where there was emergence of oxygen?
Okay, so for microbial life, they normally live in communities. First of all, they're although they are single celled, they always live like together. And then what we normally see is, well, at least with current organisms, they always live in a community between different species. So like, as I said, they're always in a metabolic cycling pathway.
So does it mean that we lost archaea when we got to the great oxidation event?
No, we still have archaea.
Okay.
So archaea and bacteria is also still here,
Oh.
but then some of them combine together to form eukaryotes.
Oh.
But you can see here that like this line still like going on, which means there's a lot of archaea and bacteria like around us. Yeah.
Okay. Yes, the question here. Nice and loud. Loud as you can.
So basically, you're using the archaea found in present day to trace back to
Yes.
So how do you know there was no mutation in between and the exact you are finding now are not different from the one back in archaea, the Hadean ages and different eras?
So you're basically asking that you're not sure if these proteins present now were present then.
Changing the environmental conditions.
Yes, so that is a very good question, and that is like a current limitation of our study, because there's only so much you can do with current organisms, and from microfossil data, which are like fossilized microbes, we can't extract any
So the
protein.
research made me untrue.
No. But then I think this is the best option, that best method we have for now, because this is how we infer evolutionary histories of proteins.
Can we just take the sample of the archaea and put it in an environment so that we think was back in the day, so that we can
So that is a very good idea, and I wish someone did it, but no one did it.
It's a really good question. Maybe I can change the question slightly and say, are there any ways of going back in the rock record from a geological perspective and trying to confirm some of the things that you were speaking about from microfossils in the rock record, or don't those things preserve?
Okay, so for proteins, they do not preserve at all. Well, at least the genetic sequences, they are degraded completely. But then obviously we can kind of see from their morphology, so like depending on which organisms are staying together within these different microfossils, we can kind of imply if there was PCD, using those organisms to infer with the current organisms we have now. But that's a limitation that we have. Yeah.
Good, yeah. Lizzie? Nice and loud.
Very interesting talk. What type of environment would favor horizontal genetic transfer to vertical genetic transfer? I would imagine that vertical transfer would be more energy effective.
No, so HGT is much more effective in prokaryotes all the time. So HGT is just a very common mechanism. It happened then and it happened now. It's still happening. But then for vertical inheritance, it's more likely with multicellular organisms rather than unicellular organisms. Yeah.
And how exactly is that process?
So for HGT, there's a lot of different mechanisms. So they can either act via like viruses or through the plasmids. So there's a lot of different mechanisms.
One last question.
You showed that the programmed cellular death includes many different molecules and complicated pathways.
Yes.
I assume there must be several hormones in there. So I was wondering, to come back to the if that's visible in the geological record, is there any lipidic hormone or the molecules of this nature that could be preserved as lipid biomarkers and that we could find? And are there any possible lipid biomarkers of PCD?
To be honest, I'm really not sure, just because I'm not a geoscientist, so I actually don't know a lot whether they could be preserved. But maybe that is a good question.
Do you know if there, for example, there are some lipids that are really typical of PCD metabolism ways?
Okay, so I think that's a difficult question to answer because PCD just involves a lot of different proteins, and these proteins are again involved in other different pathways. So there's a lot of overlap. So it's very hard to actually look for a typical protein that is associated with PCD only. So yeah, I mean, you could always look for morphology, like morphological data, but for proteins, I'm not sure.
Okay, well, Sori, thank you so much. That was fantastic. You can see a huge amount of interest based on the discussions and lots of interesting ideas. So thank you very much once again, and good luck for your PhD.
Thank you.
All right, next week we stay in the Archean, actually, but talk about rocks. And we'll be speaking about Archean crustal evolution through the eyes of granites in the Barberton terrain. That talk will be given by Lawrence Robb, who's a visiting professor here and who's based normally in Oxford. So thank you all, and we'll see you next week. Same time, same place.